Hello, everyone, and welcome to A&P again this week. I hope you guys are having a beautiful Monday or whenever it is that you're viewing this Collaborate session. A little housekeeping stuff before we get started. For some of you all that are going to review this session prior to our quiz, make sure you review the concept map, and I'm going to show you exactly what I'm talking about. That concept map with all of the connective tissues on there. If you know that map, and I've also sent this to you all in an email, then you will be well on your way for your quiz this afternoon. So I'm going to share my desktop with you all, and I'll show you exactly what I'm talking about here. Okay. Here it is. So if you know this chart, concept map chart, if you know this, you will be well set. This is the only thing your quiz is going to have on it. So know this, you're good to go. All of those connective tissues, how they relate to whichever connective tissue, what's fluid connective tissue. Know this map, you're in a good spot. All right, so just as a recap for the quiz, know that map. Make sure that you show up at our normal time for laboratory. So for our lab, show up at our normal time. We will have our quiz first. We will go over our quiz. You will turn your homework in, and then we will have our lab practical right after that. Remember, at your lab practical, you have about a minute at each station. It shouldn't take more than an hour for you to complete your lab practical. And then once it's completed, it usually takes me about a week or so to grade them. So it's going to take me a little bit longer to grade them, but as soon as I get them graded, I will post them, and I'll tell you that they have been graded. All right, so I think that's all my housekeeping sort of stuff for now. What we're going to do today is we're going to keep plugging away into this chapter over the bone. I believe it's chapter 6 in everyone's textbook. Where we last left off, we were talking about some of the features of bone. As I said before, we kind of take a tour of this organ system, and then we start to look at it on a cellular level, and then we start to look at the physiology of it. So where we last left off, we were just kind of taking a tour of it and talking about how there are bones that are classified based on shape. We can have long bones and short bones. We looked at the inside of those different shapes, comparing flat bone, like your parietal bones that make up your skull, two of the several bones that make up your skull, how they have parallel compact bone surfaces, and then the inside is that diploe, those struts, kind of like spongy bone on steroids. We also talked about how long bone has a medullary or hollow cavity where the marrow fills the inside of them, and then it has those knobby ends that we call the epiphysis, and the shaft of the long bone is called the diaphysis. We also talked about the cells that make up the bones. So we said that bone is a type of connective tissue. It is a structural / supportive connective tissue. And that structural connective tissue of bone, just like any of the other connective tissues, has those three important elements. It's got a ground substances, protein fibers, and specialized cells. The protein fibers in bone, we've established, are collagen. In fact, about 1/3 of your bone is collagen. 2/3 of your bone are mineral deposits, a hydroxyapatite. And then you have your specialized cells. There were 4 types: osteocytes, which were mature bone cells; osteoblasts, which were bone building cells. Those are the ones that eventually become osteocytes; osteoprogenitor cells, which are the stem cells that give rise to osteoblast. And then we had our osteoclast, or the bone crash cells. These are the cells that help to liberate the calcium and phosphate deposits that are surrounding the osteocytes when it's needed or called for in the body. So what we're doing today is we're just kind of continuing along with that theme, and that we're going to talk more about the exact structure of compact bone. The osteon is the basic unit of compact bone. And in lab class, we've looked at several osteons underneath the microscope, where we noticed that they have this lamellae, or this kind of arrangement of osteocytes around the central or the haversian canal. That haversian or central canal is where blood vessels come through, and then out from that central canal, you have the osteocytes that are arranged in the lamellae, and they communicate with one another with those canicular cananiculi. You have perforating canals that will go into the central canal and carry blood vessels into the bone and the marrow. And then you have what we call circumferential lamellae, which will wrap around the long bone. And the job of the circumferential lamellae is just to bind the osteons together. So for any of the lamellae, they're just kind of binding locations where the osteons are bound together. So here's a picture that you're probably quite familiar with. It looks like it's just an artist's rendition of this. It looks like it's a drawing, but this is actually a compact bone under the light microscope. And if you remember, all the bone that we looked at, it had this really cool, like, black and white sort of configuration or image. Let's see where all my little pointers go. So we have our central canal. Each one of these are an osteon. Where you see a central canal, there is an osteon. The little teeny tiny, looks like hairline fractures in there, those are the cananiculi that will connect one osteocyte with another, those cytoplasmic extensions. The darker spots here, that is an osteocyte itself. The interstitial lamellae is between osteons, and then you have your concentric lamellae, which kind of forms, it kind of looks like a tree trunk that you cut down, and it forms the rings of the tree trunk, the concentric lamellae. You won't have to label anything like this for your exam, but when we talk about an osteon, you should be able to immediately think, okay, we're talking about compact bone. If I'm talking about interstitial lamella, you should think, okay, we're talking about compact bones. You don't have to label them, but you should be familiar with what these terms describe. So here is an artist rendition, an artist drawing of it, and we get a little bit more detail here. We're able to see the blood vessels that come into the bone. They kind of come in there perpendicular and then go up through the central canals. The central canals contain blood vessels, and this is how the osteocytes are able to receive their nutrients, their oxygen, get rid of their waste products. Your bone is a highly vascular structure. A lot of times when we look at bone, we think of like chicken bones or some bones of animals that we've eaten, and we don't really see anything on the inside of it because we've cooked all of those blood vessels. But if you break a bone, it is going to bleed, and this is why it bleeds, because it's highly vascular. You have blood vessels that are coming in, bringing nutrients to supply the needs of those osteocytes or mature blood bone cells. If we go on a little bit further, we see the trabeculae of spongy bone here, and when we talk about spongy bone, there are some notable differences between spongy bone and compact bone. So the structure of spongy bone, very first important difference we want to make note of is that spongy bone does not have any osteons. So that whole setup we just talked about there with the cananiculi and the osteocytes branching out and communicating with one another, and they're bound by the lamellae, and osteons are bound together. Those osteons don't exist in spongy bone. Even though we don't have that unit of compact bone, we call it an osteon, we still have osteocytes. So there are still bone cells in spongy bone. They're just not arranged as osteons. The matrix forms an open framework. So what we call that open framework is the trabeculae. There are no blood vessels that go directly to the trabeculae, but it is still able to get nutrients, and you still are able to get rid of waste and do all the things that you would need for that blood vessels would provide. The space in between that kind of lattice work of the trabeculae is filled with red bone marrow. The job of red bone marrow is to form red blood cells, and we'll talk a lot more about this in A&P too, how that whole process works. But big things to remember: the red bone marrow forms red blood cells. It also supplies nutrients to the osteocytes, so it's able to get glucose, sugar, proteins, whatever it may be that the osteocytes require, as well as oxygen. And that red bone marrow does have blood vessels. So there is still a vascularization that happens, that there are still blood vessels that come into this area. It's just that the trabeculae themselves, unlike compact bone where we had the blood vessels coming up through that central canal, the trabeculae themselves, the struts themselves of the osteocytes, don't have a direct blood supply, but surrounding it in those spaces, filling the trabeculae, those spaces filling that kind of lattice work of spongy bone, you do have blood vessels. The yellow marrow is in some bones. Spongy bones holds yellow marrow, and it's yellow because it stores fat. So the job of the yellow marrow is to be fat storage or energy storage. Any place that you think of fat being stored, I want you to think of energy storage, as well as it can provide insulation and cushion, but for the physiological needs of the organ system, the yellow marrow or anything that's lipid stores energy. So bones like your femur are considered weight-bearing bones, and they're able to transfer the weight from your hip joint to your knee joint. And the way that your femur bones are aligned in your body is that they don't go straight up and down. A lot of times when we look at a skeleton, we see I'll just draw a little stick figure here. And there's our little stick figure guy, and there's his little stick figure leg, down to his little stick figure foot. So a lot of times we imagine that the bones go straight down, when in reality, for those weight-bearing bones, I'm just going to draw this lower half of our little stick figure, they kind of do one of these things, not quite this exaggerated. And then there's our knee that comes down, and then here's your pelvis. They're going to kind of come in a little bit at an angle, and they're able to do that. And the reason it's designed that way is so that it can make sure that the weight of your upper body is going to be that all of that weight gets transferred equally on all parts of the knee. So not one part of the knee is going to bear the brunt of all of your weight, so that it's able to dissipate that weight evenly across the knee joint. So on the lateral side, right here, we have tension that has been developed, and then on the medial side, on the inside of it, then we have compression, and that kind of interplay of tension and compression allows for the weight to be transferred from your hip joint to your knee joint without there being all of the weight just on one part of that knee joint. If it were, we'd have a lot more knee injuries just from walking around, not just from doing athletics, but from walking around. We would see lots more injuries to that knee joint because there's so much weight on just one part of the knee. So here's a pretty picture, a much better picture than what I tried to draw. That body force is going to come to the epiphysis, and then we notice that as that weight is transferred down, that compression on the inside or the medial aspect of the shaft is distributed evenly along all parts of that knee joint itself. So it's not that any one part of the knee has to bear the brunt of all of the body weight. Compact bone is covered with a membrane that we call the periosteum on the outside. The periosteum, and remember peri means around, and anytime you see osseous or osseous, that's bone tissue, so around the bone. It's going to cover all your bones except for those parts of the bones that are in a joint capsule. And at the end of this set of notes, we'll talk about different articulations and different types of joint capsules, synovial joint capsules specifically. What that periosteum is made of, it has two aspects to it. The inside layer is all cells, it's a cellular layer, and then the outside is more fibrous. There are fibers that come from the periosteum. They're collagen fibers, and they completely connect with the collagen fibers in the bone. So as a result, that periosteum has a nice firm attachment to the bone. Those same collagen fibers that connect the periosteum to the collagen fibers in the actual bone itself are the same collagen fibers that will attach to the tendons or the ligaments. So it allows for a very firm attachment for the muscles and for the other bones that are attached via the ligaments. So ligaments attach bone to bone, tendons attach bone to muscles. So these perforating collagen fibers, they allow for a very strong connection from one bone to the next, although allowing some flexibility. So jobs of that periosteum, and remember right now we're just taking a tour of some of the highlights of the skeletal system. We talked about the cells, we talked about how much collagen is there. Now we're talking about other places that we'd find that collagen. We'd find it in the periosteum, we'd find it in the actual bone itself. It makes up about 1/3 of the bone matrix itself is made out of collagen fibers. We even talked about the alignment of the bone. So all of the, just to kind of keep where we are. We're just taking a tour. We're just taking a tour and looking at all these different structures of the skeletal system. Now, jobs of that periosteum is that, one, it can isolate the bone from other tissue. So that covering around it keeps other tissue from interfering with it so much. It can also provide a route for your nervous and circulatory system supplies. So nerves and blood vessels go through the periosteum. And then because of that intercellular portion of the periosteum, remember there are two parts to it. The part that closely touches the bone, that's the cellular level, and the outside part of it, that is a fibrous layer. Because of that intercellular layer, the periosteum can also participate in bone growth and repair because of those specialized cells. So if we were to look at periosteum on a long bone, remember the knobby ends are your epiphysis here, and this is a humerus bone that we will talk a lot more about next week when we start going over the appendicular skeleton and musculature. If we were to take a cross-section of that, we'd notice that here we have spongy bone happening, and if we were just to look at the outside and peel back that periosteum, we'd see that cellular layer. So all these little pink weird shaped things are representing cells. This over here is still our bone. The light bluish sorts of hair-like fibers, those are representing collagen. And we can see how the collagen from the periosteum completely connects with the collagen that's found in the bone. Compact bone is covered with that membrane. On the inside of compact bone, we have the endosteum. Endosteum is an incomplete cellular layer that lines the medullary cavity. We also find endosteum that covers the trabeculae of spongy bone, because if you remember, spongy bone and compact bone, they aren't actually structured exactly the same way. Spongy bone doesn't have any osteons, compact bone does. Spongy bone doesn't have the trabeculae don't have blood vessels that come directly to them. They have red bone marrow that has blood vessels, whereas osteons have a direct blood supply through the central canal. We also find the endosteum lines the central canal itself. Very important aspect of the endosteum is that it contains osteoblast, osteoprogenitor cells, and osteoclast. So 3 out of those 4 different types of bone cells are found in the endosteum. So that's why we call it an incomplete cellular level, because it doesn't have any osteocytes, which is okay because we have osteocytes in the osteon of compact bone, and we also have osteocytes that make up the trabeculae of spongy bone. Because it has osteoblasts that will eventually differentiate into osteocytes and osteoprogenitor cells, which are the stem cells for osteoblasts, it participates in bone growth and bone repair, this endosteum, because it has those cells that are responsible for regenerating the bone. All right, so now we're going to kind of start switching gears a little bit, and we've had a nice little tour, and we talked about all these different elements that make up the structure of bone. Now we're going to start to talk a little bit more physiology. How does this stuff all work together? So remember, this is from an older textbook, these page numbers, so your page numbers are going to be a little bit different, and my office has been completely evacuated, and I didn't rescue my A&P book, so I don't know exactly which page numbers these are, because my book is somewhere hopefully being demolded, but we'll see. So in your textbooks, in your new books, look for the portion of your book, the headings that talk about bone formation and growth. So that's where you want to be in your textbooks. So bone development, your bones, for some people, not everybody, are going to grow until you're about 25. For ladies, because we start this fancy thing called our menstrual cycle, we usually stop growing before our male counterparts. Usually once you start your menstruation, you only have a little bit more to grow. You don't grow a whole lot after that. For males, males have a longer growing season, if you will, than females will have. So people that reach this 25, usually there are males that are still having their bones grow. And for a lot of females, there are some females that are still having some bone growth, but usually we don't have much happen. But for my brother, for example, I stopped growing when I was in the 8th grade, but my brother was much shorter than me all through high school until he got to be about sophomore or junior, and he just grew like 4 inches in the summer. I went away to college, I came back, he grew like 3 more inches. Even when he went to college himself, he grew like 2 or 3 inches in college. So he's like 6'3" now. So he started off much, much shorter than I did, but he was one of those people that he continued to grow until he was about 25 years old. He would still grow a couple of inches. So human bones, they do have an ability to grow longer, if you will, until about age 25. The process of bone formation we call osteogenesis. That is the process of actually forming the bone. Now, the process of replacing other tissues with bone is ossification. There are 2 basic types of ossification that we're going to talk about. We have endochondral ossification, and we have intramembranous ossification. Those are the 2 processes that we're going to discuss. So when we talk about ossification, we're also going to talk about this process of calcification. Calcification is just not designated to bone growth in a longitudinal manner where the bones get longer. It's just the process of replacing that osteoid matrix around an osteoblast with calcium deposit. So the process of depositing calcium salts is calcification. So it occurs during ossification, and it can happen in other tissues too. You can have calcification that happens in other tissues and other different organs as well, but we're specifically focused on bone development. 2 types of ossification: intramembranous ossification, which happens with your flat bones, and then endochondral ossification, which primarily happens for your long bones. This is the portion of the notes where we're really talking about physiology. How does this process work? And anything that is a physiologically based concept that we cover in class is fair game for a short answer question. The process of endochondral ossification, I've simplified it into 6 different steps, and intramembranous ossification has 3 steps. There's a very good chance that on your exam you will have to explain the ossification process for intramembranous and endochondral. In your textbook, it's going to have, they don't put it in 6 steps. I think it might be 9 steps or 7 steps. I'm not exactly sure. But the way that I've outlined it for you in your notes is that I've kind of condensed it and made them actual steps in and of themselves. So if you want to learn the 7 steps or the 9 steps that are in your textbook, that's perfectly fine, because it's still going to be the same answer. It's just the way that I've divided the steps up, the way that I've organized them is a little bit different, I guess I would say. I thought it was more streamlined. So with endochondral ossification, it's important to keep in mind that your ossified bone begins as hyaline cartilage. So once upon a time, when you were a uterus in your mama's uterus, when you were a fetus in your mama's uterus, your skeletal structure was what we call a hyaline cartilage mold. By the time you're born, you've had this endochondral ossification take place for most of the parts of your bone except for at the growth plate, which is going to be right at your metaphysis, between the epiphysis, the navium, and the shaft. You still have some cartilage there. And that's a good thing. We want to have some cartilage there when you're born because you need those bones to get a little bit longer. My little baby is 11 months old and she's still really kind of tiny, and I'm going to need her to continue to grow outward. So she's got a lot of huge growth plates there because those bones need to get longer and they also need to get wider. There are 6 main steps in endochondral ossification that I want you all to learn, and they are as follows. So the steps that are in your textbook that I'm pointing to now, over here, they're the same steps, they've just been abridged a little bit. So I've kind of shortened them, condensed them, and I think I made them a little bit easier to understand. So in your first step, those chondrocytes that make up your hyaline cartilage, they're going to begin to die and secrete an alkaline phosphatase, which is an enzyme that further exaggerates the process of killing off those chondrocytes. So we want that process to happen to begin in the medullary cavity. So it's going to start hollowing out the medullary cavity and clearing out that space in there. So we're clearing out that space in that long bone. As they die, they're going to leak chemicals that will attract blood vessels to come to this area. The growth of blood vessels in a new area is what we call angiogenesis. So that alkaline phosphatase helps to perpetuate the death of those chondrocytes as they get larger and they start to die. And then there are other chemicals that are released during this process that will attract blood vessels to the area. So that attraction of blood vessels to the area is called angiogenesis. As those blood vessels come to the area in step 2 here, it's going to carry with it fibroblasts, calcium salts, and phosphate. So those calcium salts, phosphates, all of those nutrients that are required to start to build this compact bone. So keep in mind that the stuff in step 1 is still happening. You're still having these chondrocytes get larger and get bigger and start to secrete this alkaline phosphatase and attract blood vessels to this area. All of that's still going on while we begin to build this compact bone on the outside of the diaphysis. In step 3, blood vessels are now going to enter into that medullary cavity, and as they come into that cavity, they bring these fibroblasts, those osteoprogenitor cells, which have those mesenchymal stem cells, have differentiated into osteoblasts, and then eventually osteocytes when the osteoid matrix of the osteoblast gets filled with calcium deposits. So those blood vessels that we attracted in step 2 and step 3, they start to carry more of these entities that are responsible for building actual bone. So all of this is going to turn the shaft first into spongy bone. So all of this will make the shaft turn into a spongy bone, and eventually the shaft of the bone or the diaphysis will become compact bone. So we have the blood vessels. We can see some spongy bone in here. More blood vessels are coming in. Where the first place that ossification takes place, we call that the primary ossification site. Keep in mind that while the stuff in step 2 is happening, we start to see step 3 things happen. As step 4 happens, we still see the stuff that happens in step 3 also taking place. So it's not that we have step 1, it's completely over, then we move to step 2. It's a very fluid sort of process. So now we have the osteoclasts that are going to be carried by the red blood vessels. They're going to start to break down that spongy bone in the shaft of the bone. So in that diaphysis, on the inside part, so all of that there is going to be broken down by the osteoclast, because we need to kind of carve out that area in the medullary cavity. We call this the primary site of ossification. For step 5, remember step 4 stuff is still happening. We're still carving out that medullary cavity there. We're having blood vessels go towards the epiphysis. So the primary ossification site happens in the diaphysis or the shaft, and then the secondary ossification site is going to take place in the epiphyseal ends. So now that we've had ossification take place in the shaft in step 5, we're going to have it soon take place in the knobby parts of it, or in the epiphyseal ends there. So the blood vessels come to those parts of the bone. The ossification process takes place here. The one big difference between ossification in the shaft and in the knobby parts of the bone is that the spongy bone is going to stay intact. So when we had ossification happen in the shaft of the bone, notice that we had those osteoclasts come in, break down that spongy bone, and completely carve out that medullary cavity, because the medullary cavity should be hollow, if you will, except for your blood vessels coming in there and your marrow that fills it up. In the epiphysis, we don't have just compact bone. We have an outside of compact bone, but on the inside it's spongy bone. So at this point here, osteoclasts are not going to come and break down the spongy bone in the epiphyseal end, so the knobs of it. And then in our last step, there's further ossification that takes place in both the primary and the secondary sites. And at this point, we're just increasing the diameter and the length of the bone in step 6. Right here, we see there's still a little bit of cartilage that's left. And there's a lot of cartilage left in this, because we're just in step 4 in this picture. But this little bit of cartilage here, what do you think that cartilage is going to be? Do you think that's going to completely ossify in this first process of ossification as a fetus? Nope, it's not going to completely ossify. If it does, we've got a problem there, because then the bones aren't going to get any longer. So this would be your growth plate. And when they take x-rays, they can look to see how much cartilage is there, and they can kind of extrapolate that data and figure out how much growing you have to do. Or if you're done growing, if it's just an epiphyseal line. So here is that growth plate, that epiphyseal cartilage there. And as the bone gets longer, as this child gets older, its bones are going to start to have this ossification take place repeatedly, where this hyaline cartilage is turned into bone until it's just a line there and this child is no longer growing. So before we move on to oppositional growth, how does that feel for endochondral ossification? Do those steps make sense, or does it just kind of feel like mumbled garbage? If those steps don't make sense, and some of you guys, it probably makes perfect sense to you. For those of you all that are listening to the recording, be sure to write down specific questions and also read over that section in your textbook that talks about ossification and those steps of ossification. Okay, and that's completely understandable, Alyssa. I kind of expected you guys to say that it is kind of blurring together right now because it's probably the first time that you've ever seen this. So listening to it again, listening to this lecture again, reading over it in your textbook, all of that is going to help you to better understand this process, because I guarantee you you're going to have a short answer question about ossification. So make sure that you understand that process. But sometimes it just takes a little more exposure to the material, and it will help you. So oppositional growth is where the bone gets wider. So it's not just getting long in ossification where we just view that. We're primarily looking at the bone getting longer, but keep in mind that the bone is also going to get wider. So that compact bone is going to thicken and strengthen with age, going from an infant to an adult. So that bone is getting wider. That's what oppositional growth is. So those epiphyseal lines are your growth lines. When your epiphyseal cartilage disappears, usually your long bone stops growing sometime after puberty, but remember, for some people you can grow well into, at 25 in your early 20s, but for a lot of people, or some people, that you can have your long bone stop growing. The epiphyseal line is visible on an x-ray, and like I said before, that's how your physicians can tell if you've ever broken a bone, and they take an x-ray of it. They say, oh, you still got a little bit more to grow, or you're finished growing, because they can see that epiphyseal line. As your long bones mature, the osteoclasts enlarge that medullary cavity, and the osteons form blood vessels around the osteons form around the blood vessels of compact bone, and that's why those blood vessels kind of get trapped inside that central canal of an osteon. Now for our next type of ossification, we have intramembranous ossification. Your book also terms it as dermal ossification, and the reason for that is because it occurs in the dermis. Bones that undergo intramembranous ossification are bones like your jaw, your clavicle, any of those flat bones. Your mandible also undergoes intramembranous ossification. Unlike endochondral ossification, intramembranous ossification has 3 steps, so a little bit shorter process. We'll leave some steps to learn. So first thing that happens is that those stem cells, those mesenchymal cells, are going to differentiate into osteoblasts, and they will accumulate in this area in order for the ossification process to begin. The bone begins to get larger as the the specules are going to spread out from that surrounding tissue, and those specules are eventually going to bind together in order to form the trabeculae. So remember that spongy bone, or diploid, which is like spongy bone on steroids, there is what we find on the inside of those flat bones. The outside is compact bone, so there are parallel surfaces of compact bone, compact bone on the top, compact bone on the bottom. In between it, you have this really strong spongy bone. So what we're forming here to begin with is that we're forming this spongy bone on the inside. So the specules are just going to be outward growth of these osteoblasts that are going to eventually form the trabeculus. As spectrals interconnect, they're going to trap whatever blood vessels they find in there. Going back to what we talked about earlier today, we said, you know, spongy bone doesn't really have a direct blood supply. The trabeculate don't have blood vessels there, but it traps in there the red bone marrow, and that red bone marrow does have blood supply. So those blood vessels that were in the area, they get trapped into the specules because they connect with one another. So all of those extensions and expansions out of what's forming the trabeculae, those osteocytes, expanding out and connecting with other osteocytes in different parts, any blood vessels that are in there are going to eventually be trapped. Over time, the bone assumes the structure of spongy bone, and areas of spongy bone may later be removed, creating medullary cavities if we're talking about in different types of bone, like in your mandible. And this process continues until the entire area on the inside is just spongy bone. So the blood supply of mature bones, there are three major sets of blood vessels. You have your nutrient artery and vein, and they enter through the diaphysis, through what we call the nutrient foramen, and we can actually see those foramen on the bone models we have in class. Your femur is going to have more than one pair because it is such a long bone. At your metaphyseal vessels are going to be at your epiphyseal cartilage, so that's going to be a direct blood supply to the cartilage itself. And that's important because those epiphyseal cartilages are going to be where we have that longitudinal growth that takes place for the bone to get longer. And then the periosteum also has its own blood supply. And the blood vessels that go into the periosteal vessels are going to provide the external or the superficial osteons with their blood supply. And then those periosteal vessels, they're also responsible for the secondary ossification centers in the knobby parts of the bone, in the epiphysis. And here's a picture of all of those different blood vessels. We have our metaphyseal artery and vein, and we have our nutrient artery and vein that are coming into the bone, and then we have our periosteal arteries and veins. Kind of get up a little close to see those, and that brings the blood supply to the periosteum as well as to the secondary ossification centers in the epiphysis, or the knobs of the bone. There are lymphs and nerves that go into the bone as well, and that periosteum contains the framework through which a network of lymphatic vessels and sensory nerves will also be carried to the bone. So a little bit of a clinical note here. So when we've talked about bone formation so far, and that those different types of ossification and the role of those blood vessels that bring nutrients to those various parts of the bone, we were talking about how it normally happens. Heterotrophic bone formation is when we have bones that are just formed outside of the skeleton. At the very beginning of this discussion on Wednesday, we talked about how some people have a little bit more than 208 bones. They might have 209 bones. And usually those are small sesamoid bones that develop in joint capsules. That would be an example of heterotrophic bone formation, where we just have bones that are forming just outside of the skeleton. A lot of times this can happen in response to abnormal stress that's taking place in the body, or an injury or surgery, or any sort of abnormal stress could lead to this. We can find bones form anywhere in the dermis or within tendons, around joints, even in the kidneys, or sometimes within skeletal muscle. So we call it an ossification process when we have bones that form from other tissues, where other tissues are formed into bone. So chemical or physical events, as I said before, any abnormal stress can cause heterotrophic bone formation. Now, a very exaggerated form of heterotrophic bone formation is fibrodysplasia ossificans progressiva, or FOP. This is where there's been some sort of injury to the body, and then we have bone forming in places where it should not form, where parts of the vertebrae fuse together, muscle and ligaments are becoming bone themselves, and it completely compromises this individual's ability to have normal physiological processes because there's bone that's forming in all of these places where it should not be formed. So what we have here is on letter A, we have what we consider a stereotypical type of skeleton. And then over in B, we have this aggressive form of heterotrophic bone formation, where there should be individual vertebrae down here in the sacrum, and we don't see that because they're all fused together. The same thing with some of these ribs, that those ligaments and tendons and muscles, they've now fused together, and they've caused the skeletal system to have this very exaggerated abnormality to them. So any of these bones here, which they should not all be fused together or should not be formed, this should be an example of FOP. Fibrodysplasia ossificans progressiva. And this is pretty rare. You don't see this all the time, but this would be what's happening there. All right, so the last thing that we have to talk about are articulations, and I believe I'm going to go ahead and save this for Wednesday. So on Wednesday, we're going to go into Chapter 9, so whether you have the old book or the new book, it's still going to be in Chapter 9. The pages are probably a little bit off, but go into the portion of your textbook that talks about articulations. Before we go, questions, comments, or concerns about anything? All right, remember to check your emails. I sent you all an email, or check the announcements. Know that concept map or that chart for connective tissues. If you know that chart, you will be acing your quiz today. Also remember to turn in your homework. Bring your homework to lab class. Come to lab at our normal scheduled time. And then put your thinking caps on and be ready to take your lab practical. All right, you guys have a great morning, and I will see you guys in just a few hours. Study well.